structured database. So the famous structured database will be like a protein data bank. Okay. So if we go to PDB is the famous structured database. So you can see this. So you can also see this, the structured database like NCBI. The PDB is actually maintained by this RCSB, Research Collaborative Structural Bioinformatics. So this is the institute actually maintaining this protein data bank. Okay, so now we click this particular rcsb.org homepage, or you can click this protein data bank also. Okay, so this is what the homepage of protein data bank. You can see here, there are so many sub links are here. You can also see PDB. There is also NAKB, and here also you can see EMDB. So PDB IHM. So they all goes to PDB. Even you can see here also PDB. So they are considered as a mirror sites for this particular PDB database. So you can click any one. It will also goes to structured database only. Okay, so all are same only. It's like a mirror sites. So if you want to search any protein structure. Now again, I'm asking one of the student give one of the protein name. So we can search here. So anyone give a name of a protein for getting structure, if it is available or not.
Cardiac troponin.
Troponin, okay. Cardiac troponin. It doesn't mean that all the proteins will have a structure in PDB. Okay. So if it is predict or if it is experimentally proven, then only it will be available in the databases. Clear? So we can search it and we'll find out whether it is available or not. So fortunately, it is there. Okay, here it is there, is it not? So you can see that NMR. Okay, usually three techniques are recommended for uploading structures to PDB. One is NMR, another one is X-ray crystallography, another one is cryo-electron microscope. So these are the three techniques usually allowed for submitting the sequence to the PDB. Okay, so when you give a keyword, the protein structures will be listed here. And as I said before, you can select any one of this protein. As per our interest, you can see topic of the proteins and the techniques through which it is identified. So by reading that, we can select our protein of interest. Okay, so you can select any one of this protein by seeing this particular topic. Solution structure and NMR structure of a C-terminal domain. So this again shows that it's only have a particular part of the protein, not the whole protein will be there. That's what it means. So NMR structure of the C-terminal domain of cardiac troponin, the N-terminal domain of cardiac troponin. So they are giving only the domains, not the full protein. Okay, so like that we will see the topic. Depending upon the topic, we will see whether the whole protein is available or not. See here also you can see a particular domain of the protein is available. Here also you can see a domain of the protein is available. Domain. So without the word domain means it may contain the full troponin. Here also you can see troponin C. That is also a part of troponin. Okay, like that it will be there. So by checking the topic, so here you can see almost it will have a common term, troponin of cardiac thin filament. So like this, by reading the topic, we will select our protein of interest. So for the demo purpose, I will select now any one of this protein now. So here, one more thing you want to know that this is considered to be as an ID. Okay, this is considered to be as a protein PDB ID. So as I told before, if you know this particular ID, if you note down this ID, if you give this particular ID in a search box, So you will get only one protein will be listed. See now, actually I searched it. You can get only one protein. That is, it goes directly to this particular protein. That is why you have to note it down this particular ID. Usually this ID will contain one number followed by 3 alphabets. Okay, so that is what general structure of this PDB ID. Then, if you see this page, so this page will show the nature of the protein and through which method this particular protein is identified. Now you can see this electron microscope. So this protein structure is identified through electron microscopy, and the resolution of this particular protein is clear at level of 12.0 angstrom. Like, you can go through this particular page, you can see this, how many number of chains are available in this structure, what is the length of amino acids, and the source of this organism. This you can also see here. In here also it may be given mus musculus, you can see organism. And when you go down also you can see that particular organism. And then only one chain is there. And then if you go down, another protein is there. So you can see troponin T is here. And you can see here troponin C is here. Okay, so troponin C and troponin T is a B chain. It contains only 74 amino acids. Then you go down, you can see now troponin I. So all the 3 troponins, troponin I is there, troponin C is there, troponin C is there, troponin T is there, troponin I is there. So this is known as the complete troponin. You can see this number of amino acid 118. And again you go down. Now you can see some of the calcium ions. So these are the ligands. Okay, what are the ligands which are attached with this particular protein will also be listed. So by seeing this page, we come to know that what are the components which are available in this particular structure. So they are called as heterogenic compounds. Hetero. See here, you can see this DEF. So this DEF chains represents 3 molecules. One is calcium ion, another one is calcium, and then you can see some of the heterocyclic compounds like molecule is also present in this particular protein. So this is what the information you are getting from this particular page. So if I want to download the structure, you have to click this particular button, display file. Here you can see this legacy PDB format. So this is the favorable format which is used for study. So we have to download this legacy PDB format. Open display file. Yeah, now it is there. So it may be some problem in that particular browser. So this is the PDB file. Okay, that is the procedure. And this is the PDB file as we see before in the format of Notepad. You can see this is the protein file. Now you can save this file. So it is actually viewing through the browser. So if you want to save, you can save file, save as. Save pages. So now you can see the file contains the extension PDB. You can save it in a download option. Save. And you can view the file. See now the 6KLU PDB file. You can see this file first through this notepad. Okay. So it's look like a normal Word file. So in the Word file, you can see the structures given in the file format. Actual structure actually starts from this atom. So this is the PDB file, structure file actually. And you can see the file ends with the word TERF. Okay. So this is what the ending of this particular file. This part is actually the structural part of the protein. So the remaining part is actually explaining about the protein, like annotations, like before, in gen sequence we see. So header, so the name of the protein, Like that, the details, what are the chains are available. So like this part is going to explain about the protein, but only the structure, actual structure of the protein starts from the word atom. So if you see the file, when you see the word atom, so that is where actually the original structure starts. So you can see here, from the atom only the original structure starts. Then it ended with TET sequence. So this is the protein structure file. Now you can see the original structure by using different softwares. You can see now Discover Studio is the software. You can see through that software, you can see the structure of the protein. So if you want to see the original structure of the protein, you need some software. Otherwise, you can see only the text file of the PDB. Okay, that is the difference between a normal text file and the structure. See, now you can see the structure of the protein through the software. The same file, if you see through this particular software, you can see the structure. If you want to see the sequence, you can select there is a hierarchy option. Hierarchy option you can use so that you can see there are A chain and B chain and then C chain is there, and the heteroatoms. What are the heteroatoms are available? Okay, so you can see here. These are the calcium and other compounds. So these are the heteroatoms of this particular protein sequence are available. You can also change the display of this protein and all by clicking a particular chain. You can change the display pattern and all. You can right click, you can say display style. You can see the atom. See now atom is in off. So if you want to change the atom into stick, it will change the structure. So like this, you can change any form of this particular structure by using this display option or ball and stick. Anything you can change. So like this, we can change the protein structures by using different softwares. So like this, you can download and you can analyze what are the sequences are available, from which amino acid to which amino acid the protein is available. Like that, you can do all the studies by using these type of softwares. But after downloading the structure, you can change the shape, background, everything you can change by using this particular software. Okay.